Hello. Right, hopefully you've all started looking back at all the stuff we've done about ATP from the nucleic acids topic. So you should have looked at the ATP molecule, how it's spent, how it's regenerated, so the hydrolysis and condensation reaction. Look at the structure of ATP again, and also the reasons why ATP is a useful molecule. We're going to come back to that in year 13. But have a look at that as well. So why is ATP such a good molecule to use? Using a whole glucose molecule all in one go is like using a £50 note when you can't get change. You need smaller packets of energy. It's a universal currency. It's got high energy bonds that are easily broken down with a specific enzyme, but not easily reactive, so they won't break down when they're not supposed to. So lots of reasons why ATP is a really good, useful molecule. So look at that. Also, look at the things that ATP is used for. So we did do that last year. ATP will be used for mechanical work. So you did in the topic with mitosis, physically in cell divisions, physically moving those chromosomes around inside the cell, mechanical work, moving things around. If things are moved in active transport, it uses ATP. But then also things like temperature for chemical reactions. The breakdown of ATP is coupled with other reactions that require the energy. So you'll see that here as well, but there's lots of examples of that. So you should know that list as well. So chemical things, mechanical things, temperature, and have a look down that full list. So we're going to move on now to glycolysis, which is the first part of respiration. Respiration has got 4 parts that you're going to learn, and it's going to be the breakdown of a molecule of glucose to gain 38 ATP molecules. And by the time you've learnt it all, you'll be able to tally up where all those 38 ATP molecules come from per molecule of glucose. So we start with glycolysis, which occurs in the cytoplasm of the cell, and then the link reaction moves into the mitochondria. So we'll come on to that. The Krebs cycle takes place in the mitochondrial matrix, and then the electron transport chain, which happens along the inner mitochondrial matrix. And so those 4 stages you get through. And it's really when you get to the end of those 4 stages that you can look at the thing as a whole, really. And we can, like I said, add up those ATPs, where did they all come from, and look again at the properties of ATP and the ways that ATP is formed. So the first topic, the 3.1, which is about ATP, I generally don't do that first because I think a lot of the key terms in that is hard to understand until you know these processes. So respiration is a good place to start, and glycolysis is the first part of respiration. So not too bad, but it is learning a chemical pathway, so you need to learn a metabolic pathway. I'll show you on the PowerPoint as well. It's not this straightforward. In real life, it's a longer metabolic pathway, more complex with more enzymes involved. But if you see that anywhere else, this is what you need. It's interesting to have a look at how complex it can be, but in this form, this is what you need to know. So glycolysis, like I said before, takes place in the cytoplasm, and you start with your 6-carbon sugar, which is glucose. So start with the glucose. And it's broken down into these 4 stages. And these 4 stages are ones that WJC want you to know as well. So it's good to know those 4 stages, even though you'll learn the pathway anyway. So the first thing that happens is the activation of glucose. I said before that one of the roles of ATP is to provide energy for chemical reactions to occur. So ATP is broken down, and then the energy can help a reaction to occur. And the way that happens a lot of the time is that the phosphate that comes from the ATP is added to a different molecule and makes it less stable. And when it's less stable and more reactive, then the other reaction can happen. And that's what happens here. So it's like here, like a money analogy, it's like we're spending money to make money. You've got to, first of all, use up a little bit of ATP first of all. So the activation of glucose is where we have 2 ATP and those 2 ATP obviously become ADP, adenosine diphosphate. So diphosphate is di is 2, so that's 2 phosphates rather than the triphosphates of ATP, so adenosine diphosphate. And I always just draw it like this, so it's on the other side, that's often how it's shown, but just 2 there. And what that will do is, like I said before, it activates this glucose molecule, makes it less stable, more reactive, it's more likely to then continue, and it's going to carry on in the metabolic pathway. So then it's physically taken these 2 phosphates, phosphate from ATP to ADP and from the other one. So we add a phosphate to each one, to each side like that. So you've got a glucose molecule with 2 phosphates now added. So now that's more likely to react. So this is a phosphorylated glucose molecule. So obviously a glucose molecule that's gained phosphates, phosphorylated glucose, and then the next stage is that is split. So you don't have to know any sort of bonding chemical detail of that, just that is split. So then you have these 2 triose sugars here. So you have 2 triose sugars, still have that phosphate on there. And those 2 triose sugars, they go through a series of reactions here. So the first thing that happens is that they get, first of all, oxidized. Now, one of the biggest things in this topic is you're going to have to pull out the chemistry, the oil rig. For some of you, the chemists, so familiar with this, it's fine. So this does really vary, I think, in this topic. Sometimes people, some of you have to really think about it, and others, it's fine. But oxidation is loss, reduction is gain. So what's happening here is that these triose sugars are being oxidized, so they get oxidized, and that's because NAD, you don't have to know exactly what it is chemically, structurally, but we're going to see it a lot through this topic, and it's a cofactor, it's sometimes called, or a coenzyme. And what it basically does is it moves hydrogens and electrons around. So it's going around oxidizing or reducing things, or being reduced itself. So in this case, the NAD now is oxidizing this triose sugar, and it becomes reduced NAD, which is often written as NADH because it's taken a hydrogen. So it's reduced NAD, also known as NADH. Okay, and because oxidation is loss, that means this sugar has been oxidized. The NAD is now reduced. So reduction is gain. That reduced NAD is carrying electrons, and that's going to be really, really important later on when we get to the electron transport chain, which is the 4th part of the respiration that I talked about. So we're in glycolysis at the start. We're going to build up to the ends. The electron transport chain is like the big finish, and that's where this reduced NAD carrying those electrons is going to be massively important. So that's what happens there. So you get that happening twice because it happens to each of those triose sugars. So NAD, and then I'm going to put this one as NADH, it's the same thing. So reduced NAD there and there. Okay. So those cofactors are now carrying those electrons, and they've taken a hydrogen. Then what happens is that the ATP is produced. So of the first original glucose molecule, I said there's going to be 38 ATP produced. And here we're only going to produce 4. So it's not going to be a massive gain at this stage, but it's part of it. So you do produce these 4. So we have 2 there, and then 2 there. And yet this time ADP being turned into ATP. So this is now forming those energy bonds. And then last one, ADP. Okay. So we've gone through the 4 stages. We've got the glucose molecule is 6 carbon sugar, so I've just shown it really simply here. That's activated by the 2 ATP. They both give phosphate, and then that makes it more reactive, less stable, and it splits into these 2 trio sugars. They both do the exact same thing. They are oxidized by NAD, which is cofactor. So NAD comes along, just for now, just accept the fact that it's just a molecule that comes along and carries electrons and hydrogen. So it comes along and becomes reduced NAD, which we sometimes call NADH. Then 2 ATP are formed here, 2 ATP are formed here. And what you end up at the end, what you end up with here, these trio sugars, once all this has happened to them, become a molecule called pyruvate. So we'll write this down here. So we've got 2 pyruvate. Okay. And glycolysis is useful by itself because it is making ATP. But really what's going on here is a preparation for later stages of respiration. So it all sort of pulls together later on. And it's just a case of really learning it at this stage. What we've made is 4 ATP down here, but 2 ATP were spent. So really, per glucose, we've got a net gain of 2 ATP. That's one of the things you have to be careful with here, is that the glucose molecule used up 2 ATP here. So even though it made 4, we've got a net gain of 2 ATP. We've also got a net gain of 2 reduced NAD, because they're going to be useful later on to produce more ATP. So what's come out of this? 2 pyruvate, 2 net ATP, and 2 reduced NAD. And like I said before, if you haven't already, look back at ATP as a molecule, that reaction where it goes from one to another, the structure, why it's such a great molecule for the job it does, and what it's used for in cells. Lots of the sort of basic, it's like a list, isn't it, of things that you have to learn, of the basic things it's used for, and then learn this process there as well. And we'll move on to next time the link reaction and the Krebs cycle.